Hello, my name is Dean Penchef, and I'm the coordinator of the DISCO project here at Natural History Museum of LA County Research and Collections. And today I'm going to give you a little peek into some things that we've found in the last few years as we've been doing this project. So first thing I need to do is orient you. There are two projects going on at the Natural History Museum right now, both of which sadly begin with the letter D. One is DISCO, which has a research project that's been running for multiple years, based here at the Natural History Museum, and has research goals having to do with exploring marine biodiversity. And that's in contrast with the new Dig-In program, which has just begun here at the Natural History Museum. That's a National Science Foundation funded project. It involves 19 institutions, of which LACM happens to be the lead, and has to do with specimen digitization, not research. So I just want to make clear these are not the same project. They don't really have much overlap, except in personnel. Today I'm going to be talking to you about the research program, the DISCO program, not the Dig-In digitization project, which is quite distinct. So who does the DISCO project at Natural History Museum? It's these folks, Adam Wall in Crustacea, Austin Hendy in IP, who works on not just dead things, but live things as well. I work on the project, Janessa Wall in Marine Biodiversity Center, Jody Martin in Crustacea, Kathy Omura, also Marine Biodiversity Center. Leslie Harris in Polychaetes is a great resource and works with us in many aspects of the project. And Regina Wetzer, the Marine Biodiversity Center, really runs the show. So let me bring you up to date really just super briefly on a basic concept that underlies a lot of what we're doing in this project. It's the idea of DNA barcoding and the use of that for environmental DNA. The principle is this. There's lots of organisms out in the ocean, high diversity of organisms. All of them have DNA. They have unique sequences of DNA. And if we're clever enough to pick the right gene or right genes, we can get very short sequences, about 650 base pairs usually, from organisms out in the wild for that selected barcode gene. That gene has differences that are sufficient enough to distinguish between species, but is similar enough that there's not much difference within a species. So that's a tool we can use to identify an individual specimen. The way that works is, let's say you have a well-identified, reputably identified specimen of a particular organism. You can extract its DNA, amplify that selected barcode gene, and then sequence that gene, and then put that gene into a database, an international database of these identifying sequences collected from around the world as a reference for that particular species. So just to be specific, the genes that we use for animals, it's usually mitochondrial CO1 gene, although there are some taxa where that doesn't work well. For plants, usually chloroplasts genes and MATK. And for fungi, it's usually ribosomal ITS. So it's a variety of genes depending on the taxon. So it's not quite as simple as one gene fits all, but within large groups of organisms, you can usually use one particular short gene sequence as a barcode gene. So that's the building of the database part. So what do you do with that database? Well, if you have a critter that you don't know what it is, you can now extract its DNA, amplify the barcode gene, sequence that gene, search for it, that sequence in the reference database, and get an identification if that organism has been put in as a reference barcode in the past. Well, that's okay, but it's not actually all that exciting. We know how to identify organisms. A lot of us do a lot of that all the time here. What's much more exciting is the idea of using that technology for environmental DNA. The idea there is that everybody sheds DNA into the environment, form of hair, feces, mucus, skin cells, all kinds of things. You do it, we do it, every organism does, even in the ocean. So imagine that all organisms are shedding DNA. That DNA is going out into the seawater. You can collect seawater in any means you want, on a filter, in a container, isolate the DNA from that seawater using high-throughput modern genetic sequencing, get a list of all of the DNA sequences for your barcode gene of choice from that collected water. Do a bunch of bioinformatics on that because you get millions and millions of reads off one of these runs. Put that together with that international reference database, and you get an inventory of the organisms that have been in the area where that water was collected. So that's the technology that we're working on, and that's what we think is a really exciting application of this genetic approach to things. So one aspect that we've been working on, amongst other people, is building that library. And we acquire specimens from a variety of places. We collect some of those specimens. We do some field collecting, mostly on shore near here. We've also hosted larger events, and the largest one of which we did was the Los Angeles Urban Ocean Expedition in late 2019, where we pulled together about 20 taxonomists from around the country who all volunteered their time to come work on this, worked through thousands of specimens. We had multiple collecting modalities. We had ships donated by sanitation agencies, bunch of divers in the water collecting things, and again, thousands and thousands of specimens were processed to help augment the local Barcode library. So some of the specimens we work on for building this library come from our partners. We have a number of partners we work with who are able to supply us with specimens, notably and sort of spectacularly, the Ocean Exploration Trust, which runs the exploration vehicle Nautilus, which runs deep sea remote operated vehicle systems, and they are very happy to give us specimens. We've also worked extensively with the Southern California Coastal Water Research Project, SCORP, and its participating sanitation agencies. As it turns out, the sanitation agencies in this area do more biological sampling than any other agency in the ocean. So they're a very extraordinary resource for specimens and highly reputably identified specimens, because that's their job, is to inventory those specimens to detect the effect of water quality changes on the environment. So how have we been doing? Well, here's a graph of specimens we've collected. We've been working very hard. You can see the big uptick at Los Angeles Urban Ocean Expedition in late 2019. And so some numbers from that. We've had about 7,000 or so sequences. We'll get another 1,500 or so from Urban Ocean. And of that, that constitutes on the order of 700 or so families, about 1,500 genera of marine organisms. So as you can see, the diversity is very high, which we knew and expected. What happens to these sequences? They go to BOLD, the Barcode of Life database. That's an international database hosted in Canada specifically for these diagnostic sequences. And as you can see, when we upload a specimen there, it gets uploaded with full data, collection data, a photograph or photographs if we have them, locality data, collection data, all of that accompanies the specimen record there. And then also the sequences get uploaded there. Here's a graphical representation of the sequence for this organism, and also the trace files from the sequencing so people can double check the accuracy of our sequence calling. So that's where the library gets built. That's where our specimen sequencing goes. So what's come out of this? Now that we spent some time building that library, what do we get as part of the DISCO program? What have we been able to produce in the last few years? Brief list there, the kinds of things I'll cover: taxonomic publications, eDNA publications, some initiatives that we're involved in, and eventually outreach. One of the things I want to mention just before I launch in here is that until I get to the outreach stage, each slide here that I'm going to show you constitutes one or more publications coming from the project. So here we go. So the first thing I'll mention briefly is taxonomic publications. Obviously, when you're working with thousands of specimens, some really interesting taxonomy comes through, and we have published several papers on the expertise, primarily Regina Wetzer in our lab, on isopods. I'm not going to talk much about that because that's kind of conventional from the museum taxonomic perspective. But we've had some successes there. We worked in collaboration with the ports of Los Angeles and of Long Beach to do a pilot study for applying environmental DNA to the port biological survey systems that they do on every several years. They do large-scale biological surveys. We partnered with them to do a combined project where they did their conventional biological surveying at the same time that we did environmental DNA surveying. Here we are picking up the seawater from shipboard at that time. So we certainly generated a report for them. Also coming out of that, of course, is a published paper. And the upshot, the sort of interesting upshot from that work, the most useful takeaway is, number one, the eDNA did recover all of and we're focusing here on fish species that the port survey also uncovered in the same day, same place, same trawls. It also came up with dozens and dozens of more species that are very plausibly and reasonably found in the port as well. The other part that was interesting, and you can see this on the plot on the left, which is a multidimensional plot summarizing species differences between localities that we surveyed. And what you can see is clustering. So the color groups were individual trawl length surveys, the different groups scattered throughout the port systems. And what you can see is that there is spatial structure, there is clustering for eDNA within the port, which was not a known answer. It is entirely possible that the water is well mixed enough that you're basically one cup of water is like any other cup of water in there as far as eDNA is concerned. That turns out not to be the case. So it can be used in relatively fine-grained kinds of surveys. So that was a really nice, interesting result. We also are using eDNA in semi-terrestrial systems, the fairy shrimp vernal pool systems. And in that system, we're looking at it as a tool to aid in conservation. A lot of species in vernal pools, which are these temporary rain-fed puddles that are a very endangered ecosystem in California, because we love to build houses where vernal pools usually are found. There are lots of species in those systems that are now endangered because they're specific to those systems, and the systems are being destroyed. And so there's a lot of concern and from conservation perspective and a lot of state conservation interest in that. The unfortunate aspect is that to study those organisms, you have to net them, capture them, and that kills them. So it's very difficult to study these endangered organisms without damaging their populations. So we're looking at the eDNA as a great tool for doing non-invasive, no-take surveys of these kinds of habitats, and are getting some very good success there. I'm going to speak no further about that because, as it turns out today, Adam Wall is going to be addressing that as he talks to you about what's going on in the crustacea section. So moving on, larger scale project that we're involved in is the CalEDNA project. And that's a project that started out of UCLA, but involved a number of UC system organizations, and was a multi-year program with the intent of really trying to take genetic technology and apply it to conservation issues in the state. One of the factors that they really focused on was the promise of environmental DNA. And so that's where we came in on this project, was helping out with mostly the coastal section of their eDNA investigations. So it was an interesting project in that it very explicitly also was wrapped around the idea of using citizen science or community science as a participating organization in terms of gathering these environmental DNA specimens from multiple habitats all across the state. Some of the outcomes from that are being published, and this one covers some predictions about different landscape types across the state based on environmental DNA analysis. And this was an interesting coupling of remote sensing data, satellite-based and aerial-based sensing, to do habitat classification, and then using those data and augmenting those data with the eDNA data. And what you can see in the lower, you can see the sampling sites on the maps on the top and some of the diversity of organisms. On the lower right, the two maps on the top are the ones to look at. So number one is that these remote sensing and eDNA surveys did reproduce pretty well the qualitative classifications that we have done for decades on these landscapes. And those are summarized in those bottom three maps are traditional classifications of the landscape types across California. And you can see the colors down there match in pattern the colors on the two top maps. The two top maps are interesting. The left one is the data classifying the landscapes based on just the satellite data, and the one on the right is when we've applied eDNA data as well, and we can see a much better, much tighter classification, much less variance in it based on incorporating the eDNA data into that. So that was an interesting piece of work that came out of that CalEDNA project. Another sub-project of CalEDNA was a coastally oriented project, and that was a really interesting comparison of iNaturalist community science observations with environmental DNA observations at the same site. This was done at Pillar Point. Pillar Point is a coastal area in the San Francisco area, right near Cal Academy, where the iNaturalist group is founded. And folks there have been taking community science groups out to this Pillar Point site for years and recording observations using iNaturalist there of intertidal marine organisms huge bias towards the beautiful nudibranchs that are there because the two folks at Cal Academy who do this love nudibranchs. So probably every nudibranch there has been photographed 5 times, but that's a side issue. The comparison we were able to do was compare in the same study area the iNaturalist observations with the environmental DNA observations that we did in conjunction with Cal eDNA. And the upshot there is that there is complementary findings from the two. There are certainly situations where, as you can see, for instance, in the top right, that plot shows the bay ghost shrimp, and that turns out to show up all over the place in the eDNA. Lots and lots of hits on eDNA sampling sites, almost never shows up in community science observations. Contrast that with right below it, the barnacle observations right below. Barnacles always show up in community science observations. They're sitting right on top of the rocks. Everyone can see them. Turns out they didn't trigger very well in the eDNA results. They very seldom showed up in eDNA. So that was one of the interesting things, that to find out that although you get very often good overlap between observations, sometimes these technologies, like any sampling technology, can complement each other's results, not just be duplicative or replicative of each other. So a larger scale initiative that we've become involved in is a group effort of the Western Association of Marine Labs. This is, you can see the map there, the localities of all the marine labs along the West Coast that have banded together into this group. And that group, primarily actually on the basis of our project, decided to launch an initiative across all of the marine labs to try to start creating reference barcode databases for organisms up and down the coast, with a view to trying to figure out how much of the similarity between species is really similar. We tend to use the same name for similar looking things all up and down the coast, but as you can imagine, there's areas of genetic isolation, so we probably have many, many more species on the West Coast than we suspect. So part of this project was the idea of trying to get localized libraries of diversity from both a genetic and morphological point of view to explore what kind of variability there might be up and down the coast in the marine invertebrate coastal system. It was attempted to 3 groups: ampellicid isopods, talinid bivalves, and spheromatid isopods. We really only had success with the isopods. The not success was not genetic. It was trouble with people being able to get out and collect the specimens. So we're in the final stages of a manuscript on the isopod results coming out of that. And the intent on this, because this project, as you can imagine, kind of came to a halt during COVID, is to re-up this, and as we start emerging from COVID, and get folks better oriented to doing these collections and getting a better picture of the genetic and morphological variability for a series of species up and down the coast. On a national scale, we're involved with the US Ocean Biocode, which is kind of taking that same West Coast idea, but doing it on a nationwide coastal basis. It's a project that's proposed to be part of the UN Decade of Ocean Science for Sustainable Development and the Seabed 2030 Initiative. This is being led out of the Smithsonian, actually. We have a strong relationship with Smithsonian there. And this is a poster publication on that project, which is in its early stages. The idea being to get a unified approach to getting barcode libraries set up all across the country. One step up, global initiatives. We're also involved with the Earth Biogenome Initiative. This is an initiative that is ultimately destined to try to get a full genome sequence for each species on the planet. The idea is to start with 1 per family, then work down to 1 per genus, and eventually 1 per species. Interesting and ambitious goal. Obviously, the question of species and specimen acquisition becomes a really interesting one for this, and that's one where we're working very closely with them, because we're one of the groups that has been spending the last few years trying to explore the ocean and collect specimens with a view specifically to genetic sequence data. This also factors into the Global Genome Biodiversity Network, which is a group, an international group that is creating a network of genomic samples, essentially doing the bookkeeping and making public the existence of genetically available samples. And on our urging, we were able to convince them that marine specimens preserved in alcohol were actually tissue samples, not just things that were frozen tissue, but alcohol preserved specimens also now qualify. And so we're participating in that project as well. So moving on to outreach kind of things, we've done a bunch of different sorts of outreach. One of the things we've done is a number of internships, one of which a program that we ran consistently for a number of years, the Advanced Research Internship Program, took groups of high school students and brought them through DNA barcoding experience with a view to learning about eDNA and lab technology and biodiversity in the ocean over a 2-week period. And we did that consistently for a number of years with a number of students each year. We also did very extensive outreach at the LA Urban Ocean Expedition project. This footage comes from the one day there where we invited the public to come join us, which turned out to be much more successful than we expected. The folks who came were really interested. Many of our community partners came out and brought aquarium specimens like this. This is not a specimen we picked up from Urban Ocean. And we worked with the folks, giving them demos and exposure to the kind of work that we do in the marine environment. So that was a really fun day. Another project we've done more recently is this Educator Workshop this summer just now, June 28th to July 2, called Dive into Marine Biodiversity. And that project was a week-long program for STEM teachers, all of whom are in a program at Cal State Dominguez Hills, Kamal Hamdan's program. So these are all in-service teachers, some of them just barely in service, but a range of expertise. Also with us during that week was a pair of artists from CSUDH who are doing a project funded by on the Pacific Standard Time program, and working on brackish water and its implications for both people and people's interactions with the environment, with the idea that brackish water is neither fresh nor salt. It's a mixture, it's in between, and that has some interesting implications. So with these folks, we spent 5 days with them. We took them out on research boats. We took them out to the Cabrillo Aquarium, took them out to the Alta Sea, all with the intent of showing them marine resources that are available to them as teachers in this area. And finally, as a capstone, we got to take them out on the tall ship for a cruise on the last day. Here's some photographs of them on a couple of on the top on the research vessel, seeing how marine specimens are very often collected using trawl and other types of techniques. And on the bottom, we took them off to the intertidal and looked at the intertidal at Point Furman, and also brought them to the Alta Sea facility in San Pedro, a very large marine-oriented education, business incubator, and research operation there, and were able to expose them to a number of activities that day. Notably, it was a really wonderful address by Bill Estrada from here at NHM on the history of the coast of Los Angeles and the social implications and social actions that have happened as Los Angeles has grown in conjunction with the coast, and the people of Los Angeles have interacted with it. We also had the fun opportunity at that Alta Sea day to christen our brand shiny new remote operated vehicle. This is an underwater camera and specimen acquisition platform, and she was named the Sarah P. Monks after a very early marine biologist here in the San Pedro area. At the just dawn of the 20th century, Sarah P. Monks was a remarkable and very strange but very productive researcher in the area. And we had the privilege of having the Sarah Monks christened by Geraldine Natz. Dr. Geraldine Natz is the ex-director of the Port of Los Angeles, and here you see the view from the Sarah Monks as Geraldine is christening her, and we then proceeded to launch her. So that was a really fun way to wrap up the week of exposure to the marine environment to a bunch of really interested and engaged STEM teachers. So what I've been able to do, and there we go into the water. I'll leave it sitting on the water for a while. I'll float for a minute. What I hope I've been able to do is let you know what some of the exciting things are that we're doing that have to do with environmental DNA, which is the real research direction, having worked very hard to build the library of genetic sequences, and some of the prospects for where we're going, investment and interest in conservation-related things, in survey-related materials, always in specimen acquisition, using genetic capabilities of specimens on a coastal, national, and global level as we move forward in the next few years. And thanks very much, and obviously we are very happy to answer any questions And you all know where we live at the Natural History Museum of Los Angeles County. Thanks.